Now it's my honor to introduce to you the next speaker, Associate Professor Sunny Wong. Prof. Wong is a clinician scientist and an associate professor at the Lee Kuan Chen School of Medicine, Nanyang Technological University of Singapore. He graduated from Chinese University of Hong Kong, and then he completed his PhD in University of Oxford, and he completed his clinical training in gastroenterology and endoscopy in the Prince Wales Hospital, Hong Kong. His main research interest now is on gut microbiome, investigating the host microbe interaction in digestive and metabolic disease, and exploring his discovery of novel biomarkers and therapeutics. Please, Professor Wong.
Thank you. Thank you for the kind introduction, and it's always a pleasure to speak after a great speaker, and I probably enjoyed the talk by Prof. Jay just now. But it's also great to have him setting the stage of looking at the metastatic niche. And so my talk is on the role of microbiome in the metastatic niche. And I have been working a bit on cancer microbiome, primarily more with the primary cancer, but we also know that microbiome is playing a role in different aspects of the cancer that includes its progression, but also its metastasis, its treatment. So on here, I will talk about the role on the metastasis. So part of my disclosure. So what I would do is I'm the only one that talks on microbiome in this forum. So so I would give a bit of time to talk about the cancer microbiome, and also the second part on the role of metastasis, where we do see a few new studies coming out. And so we know that microbiome is important. We know that microbiome is all around us, and they are everywhere in the body, particularly in the GI tract, where most of them are, but also on our skin, in the lung, and almost everywhere that you can find. We know that there are many of them. There were some disputes about the numbers, but we probably know that it's about 40 trillion. We thought it's 100 trillion, so that's why we always say 10% human. So, but we understand that it's probably an overestimate, and a closer estimate is probably 40 trillion, meaning that we are about 40% of human cells and about 60% of microbes. We know that many of them live in the GI tract. They are important, not because of the sheer number, but also because of the functional repertoire, the genes, the proteins that produce. So it's about 100 times more than what we can produce. So the functional spectrum, the functions that they carry is immense. And when we talk about microbiome and cancer, this is a graph that we are looking at. So for long, we have been talking about the genome, genetics, and also the epigenetics of cancer, and also surely we have environmental factor. And we always argue about which one is more important. I think I would bet that probably now is about 1/3 of the genetic that is important to cancer, and about 2/3 environmental. And then microbiome comes in, and so you can say it is partly genetics because it does have genes, it has metagenome, it is inside us, but also mind you that it is part of the environment as well. So it's in the air, in the food we take, and also, so you can say it's partly genetic, partly environmental, and surely they are important. They interact with both ourselves, the epigenome, and also the environment, and they do have big role in cancer as well. So this is what we are looking at. There are microbes that do not do anything. There are many that are actually commensal, so that they do have benefits than harmful effects, but there are some that causes diseases, tuberculosis, or the infectious disease, gastroenteritis, and there are some that causes cancer. And now there are some that actually promote cancer metastasis or change the way that we respond to cancer as well. This is a classical example, Helicobacter pylori, but we know there are more than that. This slide is before the age of microbiome, but even before we know about the microbiome, the ecological aspect, we know that there are microbes and they are extremely important for many of the cancer. So classical example is Helicobacter pylori and gastric cancer. This is a class 1 carcinogen, and this is the prime example for gastroenterologists and GI doctors. And but we know there are more than that. For example, schistosomiasis, bladder cancer, gallbladder cancer, and also a lot of the viruses that includes hepatitis virus, and also EBV, for example, in gastric, and also nasopharyngeal cancer, which are important in this part of the world. It is estimated that up to 20% of all cancers are related to microbes, so they are indeed very important. So what comes after microbiome? So apart from these primary carcinogenic cancer, we know that there are microbiome, which are microbes themselves, and they play a role in cancer. Their role may not be as dramatic as gastric cancer. You may not be able to find unique one single causative microorganisms, but we know that the ecological change in terms of the composition contributes to cancer. And for some of them, actually, they are more clear. For example, colon cancer is more established. We know that there are bacteria such as E. coli, Bacteroides fragilis. They are large family, but they are strain within them that do secrete virulence or carcinogenic factor that causes promote cancer. Fusobacterium is one classical example. I will talk more about this, but then there are long lists. I wouldn't say that they are carcinogens or they are carcinogenic bacteria, but the change has been related to the composition of the microbiome environment, and they do show changes. They associate with certain cancer. Apart from that, we know that the GI tract is important. That includes the gastric cancer. It's not only Helicobacter pylori. Studies have shown that apart from Helicobacter pylori, probably other microbes are important. Helicobacter pylori especially might set the initial stage of the carcinogenesis, but with along the cancer cascade, there are other microbes that are also important, and also esophageal cancer and other GI cancer as well. These are some mechanisms of carcinogenesis. I wouldn't go into the very detailed, but mind you that there are key mechanisms that involve chronic inflammation. So there are some that promote change the immune microenvironment that also matters in metastatic niche, and also there are some that clearly, for example, produce cancer toxin that directly chelates the DNA, causing mutations, and also some other mechanisms. So key mechanisms that you can see here. Well, I'll take colon cancer as one example. We know that there are different cancers, but in terms of the carcinogenesis and also metastasis, it is a prime example of how microbiome is important along different aspects of colon cancer. So this is what we're looking at. We all know about the carcinogenic cascade. We know about the two-hit hypothesis, and that there might be the genetic and also environmental factor that takes a long time to give colon cancer coming up from at least in the adenoma pathway. And we are looking at bacteria, fungus, and virus that are important. The key pioneer, actually, I would quote is Dr. Radi. You can see that actually the concept is not new. So the concept actually dates back to over 40 years ago. And and at that time, you can imagine that you can hardly imagine even in the early 70s, people are doing germ-free experiment, and this is an early concept that he pioneered at that time, that even compared with conventional mice, germ-free mice, if they are giving a carcinogen in a cancer model, they have less of the tumor. So first showing the importance of microbes. But the field, of course, remained quiet for quite a bit because of the technology, and it wasn't until we have more sequencing technology, metagenomic technology, that we would appreciate and find much more about the role of individual microbes in cancer. This is one that we always see. So this is the KeyMark study by Wendy Garrett, comparing the tumor and also finding more bacteria in people with colon cancer. And she actually found that the Fusobacterium are overexpressed in people with cancer, so you can visualize them in the immunofluorescent state. And subsequent studies have found that this is extremely important bacteria. There are different mechanisms that I would talk about. We did some work also to transplant the stool from colon cancer patients into animal model, and showing that they are not only the bystander, the observer, but they do have an active role in promoting the carcinogenesis, at least in the mice model that we test. So these are the microbes in colon cancer. Fusobacterium is there. You can, is there in the later part, but also there are other microbes that do play important role in various aspects. And so through the several mechanisms, for example, that through the beta-catenin pathway, through binding to the cancer epithelial cell, through modulating the NK cell through the TIGIT receptor, and so they do modulate the carcinogenesis. about the metastatic niche. So there are, if you look at microbiome and metastasis, we actually don't find a lot of paper, but there are a few key papers that we find, and one of the key area is on metastasis of colon cancer, which is very relevant. So I would spend some time to talk about here. So this is the, I think, a landmark study that was published a few years ago looking at Fusobacterium in people with colon cancer and also with the metastasis. So we know it is quite established that Fusobacterium nucleatum plays an important role in the cancer. But what we didn't know at that time was about the role in metastasis. So this interesting study, so what they found is that in cancer, so actually they travel with the tumor cell. So you found more Fusobacterium in the colon cancer tissue, but what they also found is you could find them in the blood, you could find them in the liver metastasis if you pick them up and look for them. And this is quite interesting and a big thing. So the next moment that you travel, you carry your suitcase, but for the cancer cell, when they travel, they don't carry their suitcase, they don't do the PCR test, but they carry some of the bacteria with them, and some of these bacteria are intrabacterial as well. And you can see that there are many studies that come afterwards looking at the mechanism. I would say that this is still very bits and pieces, so they look at different mechanisms. So for example, one is the immune pre-metastatic niche, how they change the immunity to allow seeding of the cell, but also there are other mechanisms, as you can see, involving, for example, autophagy, involving CXCR4, and also some other mechanisms. So we will have a look on that. So this is the original paper that was published in 2017. So what they did is they have 11 pairs of people with primary tumor and also liver metastasis, and they look for the bacteria by qPCR and also culture. So what you can see is that they are not in every of the pair. So out of the 11, you can see that 9, actually they find it in the primary tumor, and out of the 9, they find 7 that are qPCR positive on the metastatic tissue. But they were able to, for example, through sequencing, look at the composition. So there are a lot of the red, which are the Fusobacterium there. There are other bacteria as well, but Fusobacterium appears to be a key species, and they haven't really, and what also they do is they do fluorescence, and they were able to visualize some of these bacteria. So not only by molecular means, but able to see that by microscopy. So what has so how is microbiome important? So it promote a series of studies subsequently. So you can see that, for example, they were able to show that Fusobacterium did change the immune environment over there. So this group, they did look at the cancer tissue cell in the colon cancer, but they also had a chance to sample the liver tissue and study the immune niche in the metastatic lesion. So for example, they found that the Fusobacterium positive metastasis in the liver, they showed a change of the CD8 cell, so suppression of the effector CD8 cell, and also with an expansion of the MDSC. And Prof. RJ mentioned that this is more important. The MDSC is linked to, for example, cancer stemness and also epithelial mesenchymal transition. So setting a niche for subsequent cancer cell more cancer cell to stay there. And also they show that it is more associated with the CD8 cell, MDSC, rather than the tumor-associated macrophage. So as a study that was published last year, there were more studies. For example, in this, they compare the TCGA cohort and also the cohort. They look at relationship between the Fusobacterium and also expression transcriptional profile. What they were able to see is that Fusobacterium do have different role in different kinds of cancer. So for example, more of these Fusobacterium in proximal cancer who are MSI, and they are associated with expression changes that link to EMT and also metastasis as well. This study that was again recently published looked at chemical formite. This is important because they show it that it comes from Fusobacterium. So that using a co-culture system, they found that Fusobacterium and the primary cancer cell co-culture, they produce a lot of these chemical formites. This is from host, but also largely from bacteria. But what they were able to show that this chemical from Fusobacterium, they enhances the tumor metastasis, this time in the lung, and if they block this receptor that sends folate, they were able to see that there's a reduction in the metastatic lesion in the lung. So, we can see that, for example, Fusobacterium reprograms the cancer cell. They're important in tumor progression, as I first mentioned, through the LPS beta-catenin pathway, but also in the middle, you can see they do have a major role in tumor metastasis as well, through various pathway modulating the immune, changing the EMT, and also providing the environment for the seeding, and also several other mechanisms that we have shown in the slide, but not able to go into details as well. We mentioned about the bacteria and also the cancer cell going through the vascular barrier and going into the liver cell. So this is highly relevant. So and it was shown also in this study. This is not about Fusobacterium, but E. coli. We know that E. coli could be a commensal, but also some strain of the E. coli could have a carcinogenic role in cancer. So for example, in this study, what they look at is they also look at the tumor in the gut vascular layer and also in the liver. So what they were able to show is that some of these E. coli could modulate the layer, and they found a marker called PV1 that indicates how loose the barrier or the fenestration is, and they were able to show that impairment of this barrier actually allows bacteria to disseminate to the liver while seeding, while going into the bloodstream and directly into the liver. So this is quite relevant. We did a study a few years ago. We looked at clinical bacteremia or positive culture of bacteria that could have come from CRC. So we looked at these CRC-associated microbes, and we found that, for example, in some of these bacteria, we know for a long time that, for example, Strep bovis bacteremia is associated with neoplasia. So similar concept. So we looked at other bacteria, and we found that these early bacteremia could be a signal for subsequent diagnosis of cancer, and that I think would make sense because, as Prof. RJ has mentioned, long before we diagnose the cancer, the seeding, the dissemination might have already occurred. This is also the concept that has been shown subsequently that in a lot of these cancer patients, there were host and microbial DNA that could be detected, and also in the tissue. So some of these tissue, cancer tissue, they do have bacteria that was found as well. So colon is one example, but also you can see here, apart from colon, breast cancer, lung cancer, and some others, they do have the bacteria that can be detected within the cancer tissue as well. So a few slides more apart from colon. So I think there were studies looking at breast cancer that ties in well because from the previous slide you see that actually breast cancer somehow is a tissue or is a cancer that were found to have elevated amount of intracellular bacteria. So for example, this study looking at breast cancer, and they found evidence of intracellular microorganisms that promotes the metastatic colonization. So this is a new study, so you can see that these are more kind of the news or that reflects on this study. But what they were able to show is that through you can imagine that there could be potential of a contamination. So they did have an algorithm that tried to eliminate and compare it with the control. But what they found is that if you look at the breast cancer tissue compared to the normal breast tissue versus others, you see an increase in the bacteria here. And this increase in bacteria could be culture and also could be related to cancer metastasis. So it appears to be more important to cancer metastasis to the lung rather than the primary tumor growth itself. And I think in terms of the mechanism, we still need to understand a lot about it, but this was the first study showing that at least the microbiome within the cancer tissue especially for the intracellular bacteria, might be important for the metastasis of breast cancer. Two papers on prostate cancer, I don't find much else beyond breast and colon, but also these two papers in prostate cancer suggesting that the intracellular bacteria could be important for the metastasis. So, for example, this study looking at comparing looking at a type of Salmonella typhimurium, and they mentioned that if it is a it can affect the prostate cancer metastasis in this experiment. And also, they in the study in the right, they compare people with non-metastatic and metastatic cancer, and they were able to find some changes. This is a correlation association studies. So other cancer, as I've mentioned, bacteria are detected in many other cancer. So breast, we have mentioned, colon, but also some other cancer. But I think the role is less understood in some of these conditions. So it could be a very timely area to go into. So summary here, microbiome do play a role in various cancer. There are evidence, I think, good evidence that they do have a role in colon cancer metastasis, especially to the liver in some of the studies that we mentioned. Could be a role in breast cancer, although there aren't so many studies, but I think this is coming in. And for other cancer, I think there's still uncharted field that people, and we could study a bit more. There are mechanistic studies, they are important because they will lead to potential biomarker, for example, PB1 that we mentioned, that associate with the gut barrier. So it could be a good marker to indicate. So we need to understand the mechanism to be able to do that, to find these markers and to target them. And there are room for research into other cancer as well. So I will stop here. Thank you very much.
Thank you, Professor Wong, for such an interesting and thought-provoking talk. Let's open up the questions for the floor.
While people are gathering their thoughts, I was hoping to actually integrate both your talks, actually, and that's actually why we were so honored that both of you could join us. This was the kind of aim for it. We've seen that Professor Makers presented that there's the FASB ligand that causes the bringing in of the T infiltrating lymphocytes, the TILs, into the tumor. And we've seen also very good evidence that the liver is this immune desert. Could it be that your microbiomes are manipulating that? And could it be that the bacteria in our gut, you've shown the same, actually one of the slides showed the same ligand activated. Could it be that the gut's is manipulating that? And that's why it's become this immune desert. The point I'm getting at is prevention's always better than cure. That if colorectal cancer is already spread to the liver, we know outcomes aren't going to be great. But potentially manipulating the microbiome, would we see a change in phenotype of metastatic disease?
I think so. I think microbiome is not everything, but this is an important part because the gut is our largest immune organ. There's a lot of interactions between the microbiome and also our immune cell, and they do play a big role in changing our immune system. So I would think so. There are evidence that manipulating the microbiome could be an effective way to enhance immunotherapy through FMT or through even probiotic or biotherapeutic modulation, as we call it now. And I think so I would think that there would be a big role in that.
I think you hit the nail on the head, Glenn, which is you showed one slide very early on, which I found very interesting. It was in the dirty rats versus clean rats, and tumors don't form in the clean animals, right? So there's different ways of looking at that in preclinical models, and it turns out that you can create a dirty mouse as opposed to a clean mouse. Now, how can you do that? So one of the models of doing that is you can buy a few mice from a pet store, and then you bring those into your clean facility, your BSL-2 facility, and you put a couple of those pet store mice in with your BALB/c or your other mice, and now all of a sudden you have a dirty mouse. And the tumors that form in the dirty mice are exquisitely different than the tumors that form in the clean mice. And in fact, how the immune system is going to respond to them is completely different. So we may be actually barking up the wrong tree by basing our therapies on doing all these studies in these syngeneic mouse models that may not replicate what's actually happening in the human body. So hopefully you will come up with the antibiotic that will change the entire tumor microenvironment of the liver.
Yes, thank you. I agree with you completely that they are important. One thing that I notice and maybe disagree is that microbiome doesn't do everything. So actually in that study by Dr. Radhi, the germ-free mice, they do form tumor as well. But compare, it is probably much less. Probably less than, less than half of it. So I would say that maybe microbiome is taking large part of the environmental factor. It's not everything. The genetic is still going to play a role. It's 1/3 of it. So they do develop cancer based, but then I think probably microbiome contribute a significant part of the environment.
I think we've got time for one more quick question, and then we'll have a tea break and a very exciting debate coming up. Yep, Sridhar.
Hi, fantastic talk. Really enjoyed it, both Ajay as well as yourself. My question is, will we see a resurgence of routine biopsies before major resections, studying, I mean, one looking at genetic markers, mutations, and the other now looking at microbiome? I mean, that's question number one. The other one is, do we have anything on big data to sort of integrate these two fields?
I think this is the concept of personalized medicine, and I think this would be the way forward. Of course, for very invasive biopsies, I think we try to avoid, but for easy one, or at least then we try to profile, I think both on the host genetic and also maybe transcriptome and also the microbiome. So, and I think this is the kind of direction that personalized medicine is going for in the future. So.
Yeah, sure, 100%. I agree with everything Sunny's saying, but the last trial I showed very briefly was the trial that we just started with combination immunotherapy for resected colorectal mets. A prerequisite of being on that study is that you have a pre-treatment biopsy so that we can actually look at what's happening into the tumor microenvironment, not only after the treatment, but after the surgery. Because without having that information, how are we going to know? I mean, otherwise, we're left with a Kaplan-Meier curve of recurrence-free survival and overall survival. We have no other tumor correlates, and the stream of molecular surgery will be not reached. There was a slide that Glenn showed, which was the BRAF mutants, the V600E BRAF mutants, which we know in colorectal cancer is basically a biologically unresectable cancer, even if it looks like a 2-centimeter solitary colorectal liver metastasis. Biologically, that's unresectable. We need to know that information beforehand. Yeah. And so, without a doubt, I think we need to have those tissues that correlate before and after to move forward.
I think oncology has set a prime example because other fields in medicine are trying to follow, but oncology has taken the way forward already in personalized medicine, looking at the molecular subtype and seeing how it influences treatment. So I think that would be the way forward.
Okay, so it remains for us to thank the great speakers and for the session and close the session.
Thank you.
So we started a bit late, so we're running a little bit behind. So let's say 15 minutes for tea, which will just put us 5 minutes behind then. So we're back in 15 minutes, that's 11:05.